Hello everyone, and thank you for attending the Biotech Support Group Protein Enrichment webinar series. My name is Matt Keurig. I am the Business Development Director for Biotech Support Group, and I will be presenting to you this webinar today. This is the third part of our Protein Enrichment webinar series. The first two are recorded, and this one will be as well, and can be viewed from the Biotech Support Group homepage. In today's webinar, we will briefly describe the company and how to navigate the company's website. Then we will talk about specific products for albumin depletion, urine protein concentration, and glycoprotein enrichment. The company began operations in 1997 with the mission to provide proprietary genomics and proteomics sample preparation and enrichment reagent kits. We also provide specialty contract research, protein purification, and surface immobilization, and have a partnership with Profact Proteomics in new functional proteomics strategies. Primarily, we supply enrichment and depletion reagent kits for a variety of applications, including urine proteins, albumin, hemoglobin, viruses, glycoproteins, kinases, cyclic nucleotide phosphodiesterases, and lipids. All these products have these collective features. They are simple bind, wash, elute, spin filter protocols with no requirement for specialized instruments or HPLC. They are disposable, cost-effective, with no column regeneration or cross-contamination. They all have mild elution, which maintains the tertiary and quaternary structure necessary for measuring functional or enzymatic activity, making them ideal for both biomarker mass spec proteomics or functional proteomic investigations. All of the surface chemistries are adaptable to high-throughput 96-well formats. So as I mentioned, we'd first like to go through how to navigate the Biotech website and to see what useful information you can bring from it. So when you go to our home page, you'll see on the right-hand side of the page a news and events section. This is a good place to look for new product introductions, and we also try to keep abreast and list any particular new journal references that come up regarding the use of our products in the field. If you go to the products page, you'll see all of the Biotech Support Group products listed on this one page, categorized into proteomic sample prep, silica affinity matrices and chromatography media for proteins, our functional proteomics platform called Serophile Prospector, and a number of different DNA and genomic sample preparation products. If you click on any of these products, you'll go to the product page, and I'm going to use Kinases lipid removal as an example. On the product page is a brief listing of the product features and advantages. And if you go down here, if you scroll down, you'll see the ordering information and prices for the products, and you'll see a PDF page which gives you the product sheet, which gives you all of the protocols for using the product. And here you'll see the references. And we try to keep this as up to date as possible. So this will list all of the journal references where the product was used. On the application section, you can click on the link, and it basically what it does here is takes the references and describes in brief how the product was used in the various references. So it gives you a little overview of how the specific application for that product in that reference was used. And then finally, we also list the related products for similar work. And we also have a contact sheet at the back if you need to ask questions. You can get in touch with us by phone or email. So I'm going to close out the website and get back to the main focus of this webinar. The first area we will discuss is albumin removal. As this is by far the highest abundance protein in serum, in most proteomic application, there remains a need to simply and economically remove it. We provide two products for this purpose, Albusorb and Albuvoid, each having a different chemistry and strategy towards removing albumin. While we often get asked which is best for a particular application, in many cases there is no best answer. For these situations, we recommend the Albutrial kit, where both products are included in a trial size format. Albusorb comes from a class of solid phase or surface-based elastomeric polyelectrolytes, where the selectivity for albumin has been derived empirically, combining elements of polymer composition and cross-linking architecture. The advantage of this is, unlike antibody-based columns, the product is disposable, eliminating cycle-to-cycle variance and cross-contamination. From a 25 microliter protocol of serum, the yield of albumin depleted serum protein is about 0.1 to 0.2 milligrams. Albusorb binds the albumin specifically from serum or plasma and is compatible with downstream proteomics methods such as protein arrays, 1- and 2-dimensional electrophoresis, and LCMS. As can be seen here, albumin is very efficiently removed from serum or plasma and has been applied to human, rat, mouse, sheep, bovine, goat serums, and presumably will work with most mammals. Within the short time that this product was introduced, we have had a number of excellent journal references for using the product, which can be viewed from our website, as we described before. The Albusorb kit comes in two sizes, with all the necessary reagents to process either 172 preps or 516 preps at a very economical price, the larger volumes being less than $2 per prep. The next product, Albuvoid, uses an opposing binding strategy, selectively voiding out or not binding the albumin. Albuvoid is derived from a similar silica-based surface library with the use of individual mixed-mode polymeric ligands. The library is designed to facilitate binding of proteins, allowing for rapid elution from the matrix without any bias towards the remaining low abundance proteins. Because of this strategy, we bind the underlying low abundance proteins from serum and void out or flow through the albumin, depleting albumin and other high abundance proteins while improving the detection of the low abundance proteins. It is not based on hexapeptides or antibodies, Therefore, it's disposable and cost-effective with no column regeneration or cross-contamination. As before, mild elution maintains the tertiary and enzymatic activity and can be used with all of the different mammalian species, including human, sheep, bovine, goat, rat, and calf, and can also be used from cell culture media containing fetal calf serum. The albuvoid matrix increases the dynamic range of protein detection. In this SDS page profile, 50 micrograms of protein were loaded onto each lane, and P represents the untreated plasma, 200 the treated plasma from a 200 microliter starting plasma sample. This lane, the 200 lane, contains the eluate from the albuvoid surface, showing virtually no albumin and a wide range of low abundance proteins. Through an academic collaborator, we have analyzed the protein content by LC-MS. Not counting the antibody and keratin protein complement, 258 total proteins were identified from a combined 1- and 2-dimensional analysis. This is comparable to both high abundance depletion antibody columns and low abundance enrichment combinatorial peptide columns. Most importantly, 3 proteins were identified that have never been reported in the Human Proteome Plasma Databank, demonstrating a unique selectivity profile distinct from other depletion methods. The albuvoid kit is available in 2 sizes, 10 preps and 50 preps. Each prep is based on 200 microliters of serum to be processed. So, as we are often asked to recommend a product for albumin depletion, this table shows how the 2 products stack up. They differ in sample size, cost per prep, and dilution. For sample-specific analytes, both can be used, but the user would have to test to see which one works best for their particular analytes. The Albusorb has a modestly simpler protocol, Albuvoid having wash and elution steps. Nevertheless, the reason Albuvoid is recommended more for broad-spectrum proteomics is that it allows for much larger starting samples, whereby the low abundance proteins can be enriched on the surface. Subsequent elution producing a much greater diversity of the captured protein content. Keep in mind, however, the protocols for both of these products can be adjusted to accommodate different starting samples or application objectives. So often there will be no easy choice between the 2 products. And for these situations, we recommend the Albu trial kit, where we include trial sizes for both products. The next product on today's webinar agenda is the UPCK kit. The Urine Protein Concentration Kit is a polymeric silica-based protein enrichment matrix designed as an alternative to ultrafiltration and solvent precipitation. It has been especially optimized for proteomic studies of urine proteins, producing a 30-fold concentration of proteins in less than 60 minutes. The concentrated proteins can be applied to all proteomic analyses, including enzymatic and functional measurements. Here are SDS page profiles from the UPCK kit from 3 different samples, lanes 1 and 2 being from normal, presumably without any pathology, lanes 4 and 5 duplicate samples from a diabetic, which often clinically present a much higher protein amount, as can be seen here. The kits are available in 3 sizes with all the necessary buffers and spin filters supplied. Our last product in this webinar is the NuGel Glycoprotein Enrichment Kit based on immobilized phenylboronic acid. This product utilizes the NuGel porous silica matrix, a foundational surface technology where virtually all nonspecific sites have been eliminated, making it an ideal support for affinity purification. The phenylboronic acid, or PBA ligand, is immobilized onto the NuGel matrix. Unlike lectins, which bind to specific saccharide residues, the PBA ligand is a well-established ligand for binding to the 1,2-cis-diol groups of biomolecules, and as such, enriches for heterogeneous sets of glycoproteins containing both N-linked and O-linked oligosaccharides. Different heterogeneous sets of glycoproteins can be isolated in both the overall protein content, as in the table on the left, and in specific protein content, as can be observed from the SDS page patterns from different tissue samples. The kit can be ordered in 2 sizes, and an easy and fast spin filter format makes glycoprotein enrichment simple, starting from 50 microliter serum or approximately 1 to 2 milligrams of tissue extract. Total protein from tissue extracts can be processed. This concludes the 3-part webinar series. The first 2 webinars can be downloaded from our website. These covered hemoglobin depletion, lipid removal and clarification, separations and enrichment products for functional proteomics, kinases, and cyclic nucleotide phosphodiesterases. This webinar will also be recorded and posted on the website. Thank you, and I'd like to answer any of your questions. Well, if there are no further questions, I'd like to thank you again, and this concludes our protein enrichment webinar series.